All right. So I'm Julia di Iulio. I am a research scientist at Human Longevity. I work in the genome discovery group that is led by Amalio Telenti. And if you have any question, here's my information that you can contact me on. So today I'm gonna describe an approach that we have used to identify functional part of the genomes using only genetic diversity and nothing else. So we actually came up with this project. It arose from a recent work that made us realize that there is a major challenge coming with whole genome sequencing, which is, which is the variant interpretation. And this is because with deep sequencing nowadays, when for each individual that we sequence, we get to see more than 8,000 new variants in average. And most of them are in the non-coding part of the genomes. So this is like very valuable information, of course, this genetic information, but it would reach its full potential if we are able to interpret those variants. So in the ideal situation, we would be able to answer the question, what kind of effects have those variants? But this is maybe still too high throughput to ask right now, at least at a genomic scale. I'm sure in a few years it's gonna be feasible, but for now it's still a little bit too high throughput. So the intermediate step would be to be able to indicate whether the variants have an effect or not, or whether they are falling in a place of the genome that is functional or not. And there have been several approaches so far that have tried to answer this question and have been summarized by Manolis Kellis in a previous publication. But here, what we're gonna try to do is to develop a complementary approach. So all of them used together would be even better, but this is just a complement of what is already existing. And we're gonna use only the intra-species information to try to identify those functional regions in the genome. Before I explain to you how we did that, I just want to bring your attention to a major confounder when we are assessing variation throughout the genome, and it's the sequence context. And this is for two reasons. The first one is that different genomic elements, such as exon or introns or anything else, they have very different specific k-mers. Their sequence is not the same. And second, the sequence itself, so the k-mers, have different variation rate. In this model that we used octamers, and I will explain in a few slides why we did so. But first, let me illustrate like how genomic elements have very different sequences. So the heat map here shows the relative abundance of each octamer in different genomic elements. So each row represents an octamer, which sequence is displayed on the right-hand side, and each column is a genomic element. So I don't expect you to read like the exact sequence of each octamer, of course, but just the overall message here is that we can see that there are some patterns of octamer that are enriched in some genomic elements, for example, here in promoter, and there might be like completely depleted in other genomic region and so on. So every genomic element have very specific sequence. Again. So now for the variation rate, before I show you how different it is across the octamers, let me show you how we actually computed this value. So we go throughout the whole genome, and for each octamer, in this example, let's say A, G, G, C, G, T, G, we're gonna extract every occurrence of the octamer in the genome. In this given example, this octamer occurred up to almost 200,000 times in the genome. So then for each one of those occurrences, we have the information for more than 11,000 whole genome sequences. So we have a total of around 2 million data points to estimate the variation rate for this given example. And then we looked at the middle nucleotide of the octamer, and we are going to compute the probability of variation of this nucleotide to vary with an allelic frequency that is higher than 1 in 1,000 alleles. Sorry. So we choose this cutoff of 1 in 1,000 because the rationale is that variants that even more rare than that, they might be too recent to actually tell us anything about constraints. So we might not have enough insight or evolutionary distance to see anything. So then this variation rate, which corresponds to the proportion of those eptamers in the genome that had a variation of more than 1 in 1,000 again. So this is a pretty good proxy. This variation rate is a pretty good proxy of how the middle nucleotide has a chemical propensity to vary or not, depending on the sequence that is surrounding this given middle nucleotide. So then, as you may wonder, why did we choose eptamers? Here's why, and this is in agreement with a paper that was recently published. We actually have seen that the longest the chimer that we choose, the best the precision rate that we are going to compute. But on the other hand, the longest the chimer, the less occurrences we're going to see in the genome. Very long chimer, they're going to occur, of course, less often than a one nucleotide or dinucleotide and so on. So this is an example with real numbers. For example, if we take all the cytosine in the high confidence of the genomes, we compute an average variation rate of 1.2%, always with an allelic frequency higher than 1 in 1,000. And to compute this number, we used more than 500 million counts throughout the genome. Now, if we increase the chimer, if we look at dinucleotide, for example, we can see that we increase the variation rate precision. Now, instead of 1.2%, we have like a more precise numbers. For example, 0.8% when it's followed by an A, while it's 8.1% when it's followed by G. So now we gain precision, but again, we decrease the number of occurrences of the chimers to compute those precision, and so on and so on. So overall, eptamers, after eptamers, like with longer chimers, we actually tend to reach a plateau for the precision. We don't gain much additional information, while with eptamer, we still have sufficient power to compute those variation rates. So that's why we chose eptamers. All right, so if we come back to the previous slides, now I can show you like how much the variation rate varies across the eptamers. So this is a cumulative distribution function showing the variation rate across the 16,000 eptamers. And the size of the dots is proportional to the occurrences of the eptamers in the genome. So what's interesting here is that we see like a very wide distribution. It starts from, it's a logarithmic scale, so it starts from 0.15% up to 15%. So there is more than 100-fold change across eptamer in terms of variation rate. All right, so now I hope I convinced you that the sequence context is really important to take into account. So when we analyze variation throughout the genome, we should definitely take this into account. And this is how we did it. We go throughout the whole genome with a sliding window. And then for each one of those windows, we're gonna extract the number of observed variants that had an allelic frequency higher than 1 in 1,000. And this is obtained with the more than 11,000 genomes that we sequenced. And then we're gonna compute the expected number of variation with an allelic frequency higher than 1 in 1,000. So this is based on the eptamer variation rate that I just described. So now, because every single nucleotide in the window is the middle nucleotide of an eptamer, we have the variation rate for every single nucleotide in the window. So now if we add up the probability of variation of each one of those nucleotides, given their sequence context, then we're gonna get the total expectation of variation for this given window. So if we subtract the expectation from the observation, we then obtain what we call the context-dependent tolerance to variation score. And this is what we're gonna use as a metric to identify functional part of the genome. So here, once we've gone through the whole genome, we rank the genome according to this context-dependent tolerance to variation score, which we will call CDTS. And please try to remember this abbreviation because it's gonna come pretty often. So we're gonna rank all the genome, and on the left-hand side, it's gonna be like the region that had the lowest tolerance to variation, and on the right-hand side, the regions that have the highest tolerance to variation. So whenever I say tolerance to variation, I always mean context-dependent tolerance to variation, just to be a bit shorter. Okay, so now because we have used so far no a priori knowledge at all and no annotation to establish this metric, we wanted to see how they match known genomic annotation. So we looked at the composition of those different regions. And this is what we see. So here we have like 10 different genomic elements family, the most common in the genome, and the 11 ones is the conglomerate of all the others that were not so common. All right, so what we can see first is that there is a wide variation in the distribution of those elements throughout the CDTS spectrum. It's not similar in all the bins. Second, we can see, for example, that in the regions with the lowest tolerance to variation, this part here, there is a big chunk that comes from exons that are from protein coding genes, which is not unexpected. But what's interesting as well is that those exons coming from protein coding genes, they're not only present in this part of the genome, at least in the CDTS spectrum. They are present a little bit everywhere, meaning that all the exons might not be as important, so which is also known, but it's nice to see. Second, we have seen that there are a lot of promoters in this region of the genomes that have very low tolerance to variation. And this is in contradiction with what is usually known, because we know that promoters tend to vary a lot because they are enriched for CPG islands. But what we're saying here is that even if they vary, they might still vary much less than they're expected to given their sequence. So they might still be like under constraint. It's just that given their sequence, they are seen as variable, even though they're less than they would be expected to, at least for a part of them. And finally, we can see that in this first percentile, there is like every single genomic element is present there, even some parts that are completely unannotated with either ENCODE or gene code or anything, meaning that there is like some interesting feature that could be learned from those regions as well. So the first question that we had was, because we have seen that, for example, exons, we have seen that they are not all located in the same region of the CDTS spectrum. So are some elements from the same genomic family more functional when they are present at the lower CDTS? In other words, are exons that are here different from exons that are on the other spectrum of the CDTS? So to look at that, we are going to use another metric that is gene essentiality. And you are much more familiar than I am with this metric. So pretty much it summarizes like how much constraint did the protein coding region of a gene have. And it has several properties. For example, the most essential genes tend to be in larger protein complexes. They are more likely to be lethal in knockout mice. They are more likely to be haploinsufficient or intolerant to truncation. And just so you know, from now on, when I talk about functionality, I always mean functionality as assessed by CDTS. I know it's a word that could be understood in many ways. So here it's as assessed by the CDTS metric. All right. So this is what we see when we compare the gene essentiality with the context-dependent tolerance to variation score. And we can see that exons that are in the region of the genome that have the lowest tolerance to variation tend to be in genes that are more essential. And just as a reminder, keep in mind that we don't expect a one-to-one relationship, because gene essentiality has one score per gene, while CDTS has like a one score per exonic bin. So meaning that even essential genes, they might have some exons that are not essential, while some non-essential genes might still have exons that are very functional or important, and so on. But the trend is definitely there. When the exons that are the most functional in our hands, they tend to be in genes that are more essential. And then we wondered whether it was the same for promoters that are located in the region of the genome that have the lowest tolerance to variation. And again, we see the same trend. So those promoters tend to fall upstream of genes that are more essential. And it turned out that it's true for every single genomic element. So whenever they are present in the region of the genome that have low tolerance to variation, they tend to be associated with, or closer, their closest gene is a gene that is more essential. And it's interesting because even the elements that were depleted in those functional regions, for example, H3 kinetylation, it turns out that when it's present, it happened to be in the vicinity of essential genes. So this brought us to our next question. Now that we have seen that cis elements seems to be like coordinated with the gene that they might be regulating, or at least that is in their vicinity, is it also the case for distal regulatory elements? To look at that, we used high-C data, and we looked at distal regions that are brought together by chromatin looping. And we tested the functionality. So we tested whether there was any coordination by looking at the functionality of the upstream and downstream anchor. So we wanted to know whether when the functionality of the upstream anchor is low, when it's high functionality, is it associated with another high functionality anchor, and so on. And we saw exactly this pattern. So the upstream anchors that had a very low tolerance to variation happened to be associated with downstream anchors that also had low tolerance to variation. And the trend was pretty neat. And this happened to be like the case for up to 2 megabases, when the loops were up to 2 megabases long. So we finally decided to ask the last question with regard to this purpose. Are we able to see a coordination between distal enhancer and the genes that they are associated with? And because it's very difficult to associate distal enhancer with genes, we use two different data sets. So the one that I just mentioned earlier, high-C data, and we inferred the association between enhancer and gene based on the distance of the enhancer and gene to the loop anchor points and the directionality of the gene. And then we also used a promoter capture high-C data from our collaborators, which actually they directly assess promoter interaction with distal regions, which makes it easier to then associate the promoter to the enhancer. And as usual, we look at whether a functional enhancer would associate with an essential gene. And in both cases, despite the difficult technicalities or the difficulties, we actually see the trend in both situations. So when the enhancer happened to be more functional, so is in region of the genome that had the lowest tolerance to variation, they happen to be associated with genes that are more essential. Overall, those results, both the cis and the distal coordination, brings like a new concept that the gene units might be coordinated. So meaning that when you have an essential gene, you're gonna have an important promoter, functional promoter, that doesn't tolerate as much variation as another one, let's say. And then there's gonna be an important enhancer as well, and a distal enhancer, and so on, like such as coordinated gene units. All right, so then we wondered whether our metrics could be used in also like in clinical settings. So could we use this scoring to help prioritize variants? And to look at that, we compared the functionality to variant pathogenicity with rationale that if a variant falls in a region of the genome that is functional, it's more likely to be pathogenic. And we did indeed see a very marked effect. So variants that were falling in the most functional part of the genome, so the region with the lowest tolerance to variation, they were indeed highly enriched for pathogenic variants. And what's interesting is that it was the case for both protein coding variants, but also very interestingly, even for non-coding variants that are falling outside of any exonic regions. So this was really really good to see. We then used another data set, which was based on manually created Mendelian trait associated variant, just to check again that this is the case. So here we are looking only at non-coding variants. And it came from a recent publication as well. And again, we see the same pattern. So the variants that fall, there is a high enrichment for a pathogenic variant in the first percentile of CDTS. So in the region with the lowest tolerance to variation. All right. So to summarize, I hope I convinced you that context matters and that we should always take the sequence into account when we look at variation throughout the genome to try to interpret a variation in within population or within species and so on. Second, we have seen a concept of coordinated genic units. So for both cis and distal regulatory elements, we did see that when the gene was essential, the regulatory elements, whether they were cis or distal, they would be, they are more likely to be functional. And finally, we were able to show that functional, as assessed by CDTS, was a good proxy for pathogenicity as well. So if you have any questions, feel free to contact me, or you can also check on the paper. It's on bioRxiv. And finally, I'd like to thank everyone that contributed to this work. So all my colleagues at Human Longevity, and also our collaborators that provided us with the promoter capture high-seed data. Thank you.
Thank you, Julia. You can stop sharing the screen just on the top. You can, yes, perfect. And if people have questions, feel free to ask. Did someone already say yes? Okay, so otherwise I will start, because I was wondering, how do you select the promoters? Sorry, maybe you said that, but I was not aware.
No, so you're right, I didn't mention, we actually use the ENCODE annotation from multi-cell, and so I used that known annotation. So the scoring metric is done without any apparent knowledge, but then we used known annotation to about the scoring metric itself.
So, and if you do this with the PLI genes, you would just look at the next promoter, which is near.
Exactly, whatever, the first ENCODE annotation as promoter, that was the closest.
The closest. Okay. It seems to be work quite well. It was interesting.
Yeah, surprisingly.
Any questions from other people? Okay. Yeah. I guess most people will see it anyway online. So thank you very much. This was a very informative hour, I guess. Thank you, Julia, for presenting. And everyone, I wish everyone a nice weekend, and see you next week. Okay.
Thank you.
Bye.
Bye-bye.